So I'd like to just briefly introduce, we have with us Colonel Dallas Hack on the far end, who's a retired colonel and director of the, retired from his directorship of the U.S. Army Combat Casualty Research Program, and chair of, and he also serves as chair of the Joint Program Committee for Combat Casualty Care from 2008 to 2014. Next to him, we have Dr. Patrick Bellgowan from the National Institute of Neurologic Disorders and Stroke, and he leads the program for traumatic brain injury there. Next to him, we have Dr. John Krystal, who is the professor of translational research and chair of the Department of Psychiatry and professor of neuroscience at the Yale University of Medicine. In the middle, we have Lieutenant Colonel Dennis McGurk, who's the director of the Military Operational Medicine Research Program today at Fort Detrick as part of the Department of Defense. We have also Dr. Justin Sanchez, who's the Director of Biological Technologies Office and Defense as part of DARPA, which is the Defense Advanced Research Projects Agency. And finally, Stuart Hoffman. Dr. Stuart Hoffman is the Scientific Program Manager of the Brain Injuries Portfolio in the Rehabilitation Research and Development Service and Senior Scientific Advisor for Brain Injury in the Office of Research and Development, the ORD. Thank you, gentlemen. Please join us. Thank you. So this morning, we would like to discuss the National Research Action Plan for PTSD and Traumatic Brain Injury, which was mentioned by both Secretary Shulkin and also the FDA Commissioner earlier today. In 2012, the President issued an executive order for improving access to mental health services for veterans, service members, and military families. And in response to this, in 2013, the National Research Action Plan was established. And this is a coordinated effort by the Departments of Defense, the Veterans Administration, the Health and Human Services, and Education Divisions branches of our government. So that's why we've invited all of these illustrious gentlemen to join us this morning. Well, you'd like to start by asking Colonel Hack to please describe to us the background around the NRAP, and we've provided to you the slides because I realize it's a little difficult to see.
Yes, good.
The screens from here. And if you need to point to any item, you can call out the number of the slide and they will show it for you as we go through the discussion.
All right.
Okay? So, Dallas, would you mind?
It's a pleasure. Thank you. I'm privileged being here today and having you all here. We go to the next slide. This just gives a bit of a summary. The federal agencies have been having an ongoing relationship and interaction, coordination among them. We had developed joint programs together and so on. But it was felt by the White House that we needed to do more, particularly in light of the problems we've all been talking about here, that this is such a major issue that it would take the resources of the nation to be able to address that. This is why he issued the executive order. On this, there were the 7 different sections within the executive order, including care was one of the big ones on that. That's not what I'm prepared to talk about today. There was Section 5, which really had to do with coordinating the research in the area. And that's what we ended up spending our time developing, that research action plan as directed by that Section 5. The next slide just gives a kind of a cover. At the 2 slides from now, there will be actually links. You can get these documents from the White House website. Let me go on to the next slide after that, which are who was directed to be part of that. You can see that there's 3 or 4, depends how, at the time it was 4, it's now 3 different federal agencies or departments that actually were pulled together to do this. However, there are actually 15 of the different federal agencies and departments actually working in neuroscience. This is the amount of effort going on in the federal government on neuroscience is phenomenal. Obviously, we were interested in the medical aspects of it, which is why we pulled the main organizations involved in the medical parts of it. But there's actually a lot of work going on in commerce and in other areas that actually is coming up with some very interesting things, I think, that we could leverage as well. But we actually pulled together just the research action plan. As you can see, it was the DOD, the NIH, VA, and at that time, Department of Ed, and that section of Department of Ed has now moved over to Health and Human Services, which is why I have it outlined like that. It's that same group of people, but it's now realigned under HHS. A process when this was issued is that we had been ongoing relationships or joint programs, those kinds of things, and this really gave it a lot more visibility. We pulled together over the course of the 8 months, we were given 8 months to have a final document prepared out of this. We pulled together representatives from each of the different federal agencies, developed a very comprehensive spreadsheet of the efforts going on, the gaps we identified, the directives that were in the National Research Action Plan, how those all aligned with it, the length of time that we thought it would take to actually address those problems and those gaps and so on that were mentioned in that. And as a result, we authored the National Research Action Plan. Stuart and I were the only ones here that were actually part of that overall team, but there are other people that were represented here were actually, their predecessors were involved in that. It was vetted through the White House and through a number of the other agencies and so on, and finally released at the end of August in 2013.
Thank you very much.
It's so much, in the field of the TBI, the whole world really woke up to the TBI part in about 2007. Interestingly enough, the PTSD part, it was Vietnam, I think, that woke the post-Vietnam era that really woke up the world to the issues of PTSD. It had been kind of going on, but it was really that whole era. So it was interesting bringing these two conditions together, which we found out there's a lot of comorbidity in the process. So thank you, and I'll pass it on to the next.
Thanks, Dallas. We really appreciate the background. And now Stuart Hoffman will walk us through a little bit of how the pieces are put together now as we move forward. Stuart, if you could describe for us a little bit of the objectives of the NRAP and how things actually are put together in the actual plan.
Okay. I guess I want to thank Cohen Veterans Bioscience for this invitation and the opportunity to speak here, and to my colleagues here, especially Dallas, who we both on the TBI side basically hatched this idea of TBI as a chronic condition, and and to my fellow partners on the NRAP right now, and the previous ones who are not here. But as for the NRAP, as Dr. Hack said, the genesis really came from the executive order and that Section 5 for research. And so what the Section 5 really laid out for us was action items that we then incorporated into the National Research Action Plan. And
we can move the slides.
So slide 70.
Yeah, actually, this is a good slide if anyone wants to download any of this information.
The links there, and these will be available.
Yeah, so this is all public information, and easily accessible. So go to next slide. All right.
It's slide 70, please.
And next. This is just slide that basically was the kernel to what we really put together in the NRAP. There was all these unanswered questions, a lot of them going back in time a good deal, but we were able, due to our collegiality and from our leadership, Dr. Woodson and Dr. Petzel, to really codify these into something like the National Research Action Plan. Next slide, please. So we didn't. What the executive order really stated was this lack of understanding about PTSD and TBI, And this lack of understanding really hampered how we could progress in prevention, diagnosis, and treatment of these disorders. And the NRAP directed the agencies, which you see here, to work together to come up with a structure to help approach these problems. Next slide, please. So these were the goals that came out from the National Research Action Plan. As you can see here, better biomarkers for diagnosis and treatment. As you heard Dr. Marmer said, that we still don't have answers for these, and we're working on them. Improved diagnostic criteria for TBI. TBI is right now classified as three injury severities: mild, moderate, and severe. All you got to do is go to the cancer literature, and you can see all the different types of classification for cancers and all the different grades. For TBI, we just still have 3. And thanks to the efforts of Dr. Hack, we now have the TED initiative, which is working on developing better classifications and better outcome measures for these disorders. For TBI, it's actually a constellation of disorders. And also looking for mechanisms for PTSD injuries, disorders following TBI. And I won't go through all these points because we're short on time, so you can go to the next slide. And out of that came the National Research Action Plan, which is a testament of how when agencies can get together, can produce a document that can shape the field when it comes to TBI, PTSD, suicide, and data sharing. Next slide, please. And so for the National Research Action Plan, one of the major things that has really brought us together is every year, and as Pat now knows, we do a review and analysis of the portfolios on TBI and PTSD, as Dr. McGurk and Colonel McGurk does for the psych health side, where we get together and we compare our portfolios and do analysis. Matter of fact, I'll get to a slide later where we actually put the research in different buckets and we actually present each other's research in their agencies. So we really get down and we understand what the other agencies are doing in terms of TBI and PTSD and suicide research, as well as data sharing. And we continue to develop processes that standardize and integrate. And I'm sorry, but as your predecessor helped to start the TBI, Dr. Mona Hicks, who was one of the original members, developed the TBI CDEs, which is leading to how we enter data into FITBIR, which Pat manages and oversees, which is a federal-wide database that's big data for TBI, and now PTSD and other and suicide prevention is now developing their own CDEs. Next slide, please. And so one of the things I know the Cohen Veteran Bioscience is interested in is helping us to provide repositories for biological samples such as brain tissue, blood, and cerebral spinal fluid. That's under the National Research Action Plan, and to archive these and try to find pathological mechanisms that are contributing to these conditions. Next slide. And I can go to the next slide, actually. And so this is what I was talking about. When we break down our portfolios and put them back together in one common bucket, we have these bins. And we can break these down into these different aspects of science, and allows us to find the areas of need as well as areas of overlap. So we can basically synergize and really improve how we conduct our research across the agencies. Next slide.
I think that
That's it.
Wraps up the NRAP part of this. Thank you very much for giving us that overview and also showing how comprehensive the approach has been and how interactive it's been integrated across all of these agencies. And so what we would like to go through now is a little bit of an update on progress by each of you within each agency, starting with you, Colonel McGurk, for the Department of Defense. And I believe that might be slide number 79. Yes, thank you.
Yep.
You could go to 80, please, if that's 79. You'll need to see my name. Although I would like to thank you for the invitation. and Ron Hoover in particular, and all the folks that were just mentioned, that we are, to be honest, we're forced to work together now. So we were aware of each other's work, and I'll tell you that's really the point I want to make from my talk, is that it used to be, I knew, or the groups knew, we each worked in the same areas. Then the NRAP forced us to then start reporting on what are each of you doing. And I will tell you where we've begun to go and where the future is we're doing it together. Because there's not enough money and there's not enough investigators, which a lot of familiar faces are here of our investigators. If we don't all work together, pool our resources, we'll get there, but I won't be alive, and probably nobody in the room will. So if we want to get there, you know, if you want to go fast, we got to go as a team. So I think that's a real message that the NRAP has pushed us to do. And then when you start to work with these folks, you realize that we're all of a like mind. So I think that's really where the NRAP has helped us out a lot. So those priorities, the ones that are highlighted are the ones in military operational medicine. So I'm going to have to stand up if you don't mind.
Oh, no problem.
So as you can see, these are the ones we work closely with the TBI because there is a lot of overlap, but predominantly we're looking at early diagnosis and effective treatments for mental health disorders. PTSD is the one that's always first mentioned, but there's a lot of comorbidity, and I'll mention that at the bottom with that last bullet. But we really are looking to understand the mechanisms. It was mentioned the RDoC approach, really trying to get at why are things working or why are they not, so you can develop better interventions as you go along, to try to get better treatments, again, on the psychological health, and really working data sharing. And I will also mention, Stu, thank you, not just data, but sample sharing, because they're so hard to get. And once we have them, lots of people can do lots of biomarker work, replicate each other's work. That's another key area I really think we need to push as we go at this together, is sharing of each other's data plus samples. What else we have? It mentions PTSD. It mentions TBI, the National Research Action Plan. It also mentions suicide prevention. Again, very comorbid with PTSD. But it doesn't mention things like substance abuse, which we do fund research in. It doesn't mention things like workplace violence, family health and well-being, and these are areas that military operational medicine we fund because they are also interrelated. Can we go next slide, please? I had 5 minutes, I went 7 slides. I'm from Jersey, I'll talk fast if that works for everybody. This just tells you some of the programmatic advances, and the real bottom line here is we're working together. Within the DoD, the last bullet had a funders meeting. So if we're going to fund it, I have a responsibility as a joint program committee chair to report everything across the DoD. But as I said, reporting other people's work was great. It was a step forward. It's not enough. We got to work together. That's really the bottom line for that slide. Next slide, please. These are just some of the scientific advances. And things I want to point out very quickly there is we know PE, CPT work, but how can we do them better? How can we do them maybe using technology? How can we do them in compressed manner? How can we do it so it will actually work in a DoD or a veterans population? Because there are restraints, especially for a DoD population. In suicide, I think it's interesting to note again, maybe looking at apps that might help in these things because they're everywhere. There's a whole discussion of everybody in this generation, they look more at their phone than they do at each other. That's a whole other topic. I think in substance abuse, we're really looking at, again, the challenges that are there and what are the mechanisms we can look at for substance abuse, whether it's comorbid or not. But generally people don't come in and say, look, I'm drinking a lot, but everything else in my life is perfect. It's all good. Nobody does that. Next slide, please. These are just some of the remaining questions. We will be doing, here's a little plug for a DoD meeting that will come up in June of this year. We are doing a state of the science meeting on the pathophysiology of PTSD to try to identify targets for drugs, for pharmaceuticals. And that's where we really need our partners at Cohen and we need our partners in industry. Because again, to put something through the FDA process, you all know this, it takes a lot of effort and it takes a lot of money. We all need to go at that together. Again, we're trying to optimally deliver things. I've mentioned that. A big problem in the DoD, a big problem in VA, and I think as was mentioned earlier, a big problem in society is opioids. And we have a lot of pain after these wars. You saw that picture of the guy with all that stuff on his back. That's actually 2 people, by the way, in case nobody ever looked. There's another person behind him carrying some of that. But that all caused a lot of musculoskeletal pain, and people were treated with opioids. And then some of them, they never were able to come off. So that's a key issue. Next slide. These are just some of the challenges. It's hard to do Better research at multiple sites, better generalizability, also more IRBs, more regulatory. One of the other things we're facing as a challenge where I think we can all work together is we have a very nice process for what do we do if we develop a pharmaceutical intervention or a material product. We know what the FDA requires. We know how to get it through the process. For knowledge products, it's a lot harder. What's the level of evidence to change a CPG? Right? I mean, we need to get there together, establish what those levels are, work there, pick out the research, and then deliver things that are going to affect service members or veterans or their families. And then we keep doing the research to keep improving it. But if we don't deliver evidence-based research, leaders will get what they can get, especially in the DoD, I will tell you that. I think the way ahead is, this meeting is the start of the way ahead. And what Cohen is doing is, again, the start of the way ahead of interested organizations working with the DoD, working with the VA, working with NIH and all the other agencies to try to go at these problems. And I'll tell you, my last bullet there is, it was mentioned by Dr. Woodson. Academically, it was my data. It was how do I advance? How do I get my publications? How do I get tenure? I will tell you, surprisingly, in DoD labs, when I asked them, what is your deliverable? If it was a material product, what their deliverables were, publications and presentations. How many publications and presentations have helped a service member or a veteran? Directly, zero. Dr. Woodson mentioned we need to change policy. We need to change clinical practice guidelines. We need to change training. We need to change education. Last slide. So I jammed a lot down your throats. These are my 15-month-old twins. This is from their birthday. So you might be like my son saying, yay, McGurk is done. Or you might be like my daughter saying, man, you just jammed a lot down my throat in just a few minutes here. But any questions, I'd be happy to take.
Thank you so much. You've given us a lot to think about, and obviously there's been tremendous progress made at the DoD. We'll come back to some of the challenges and opportunities, and I'd like to now move on to Dr. Sanchez for DARPA. I think you were going to, you don't have any slides, right?
No slides today, but that was a great introduction to what I would like to talk about. So I'm from DARPA, Defense Advanced Research Project Agency, and our focus is breakthrough technologies. And the area that we're working very heavily in is in the area of neurotechnology. So you just heard, how often do we see solutions or technologies that are actually helping veterans? The people we serve are veterans, and we put a challenge out to the scientific and medical communities, and we said, when it comes to neuropsychiatric illness, so whether it's PTS or whether it's addiction or whether it's something like a TBI, what can we do different to isolate the subnetworks of the brain that are involved in those kinds of conditions? What kind of technologies can we develop to go directly to those networks of the brain, access the information that those neurons are telling us about their condition in moment to moment, and then develop precise therapeutic interventions that can actually have a behavioral effect in their life? And when we initially went out to the community, we said, okay, who can step up to actually do this? We heard the technology doesn't exist to go to those subnetworks of the brain. We don't have the ability to read out that information in real time. We don't have the ability to kind of model that activity and deliver therapeutic interventions. So again, we need to step in. We need to do something different here. So we started off on two programs 2 years ago. One program's called Subnets, and that refers to the subnetworks that are involved in neuropsychiatric illness. And we have a secondary program called Restoring Active Memory. Both of those programs aim to, in 4 years, develop fully implantable medical devices that can go to 2, 3, 4, 5 different targets of the brain, can measure the real-time activity of individual neurons in those networks of the brain, and then deliver that closed-loop therapy for actually having an impact or an effect. And what I'd love to share with you today is that in just 2 years of doing this work, we've already been able to show proof of concept of being able to restore the ability to form and recall memories in people that have difficulty in actually doing that using these direct neural interfaces. And then we also have examples of people having a reduction in their anxiety level by using this kind of an approach. Again, measuring the circuits related to anxiety and then delivering a very targeted kind of mechanism. So, the real take-home message in all of this is that an agency like DARPA can step in and can set the challenge out there, can set here's the prize, here's what we're actually going after, can rally the scientific community around a very focused therapeutic intervention. What we would love to do is, once we're successful and once we show what is possible in all of this, is work with all of you to get the technology out of our purview, again, and showing that proof of concept, and actually transitioning it into patient populations that can really use it in the best way possible.
Thank you, Justin.
Thank you so much.
It's very exciting to think about the potential of neuroelectronic interfaces. We're always thinking in very traditional terms about pharmaceutical medications and drugs and molecular targets, but I think DARPA, not atypically, has just sort of gone around.
Find another way around.
Right.
That's the approach. Yeah.
Fantastic. So Dr. Bellgowan, who's at the Neurologic Institute for Stroke, I think the NIH has done a number of efforts, and you're representing today, I think, both sides, NIMH and NINDS activities. Would you like to?
Sure. So NIH is made up of 27 institutes and centers, as you know. For TBI, 12 of those are invest somewhat in TBI. NINDS, which is Neurological Institute of Neurological Disorders and Stroke, we have about 70 or 80% of the TBI portfolio, and it's a fairly sophisticated portfolio in that it's complicated in that it's multidimensional. So it goes all the way from basic disease research to clinical trials, and it goes all the way from the mild concussive injury to the most severe injury. The other key to remember with NIH, and one of the great things that NRAP allows us to do, is that NIH is very investigator initiative driven, which means we ask you to tell us what the best ideas are. We bring them to peer review. We have you guys decide which the best ones are and fund them. So we don't have, for instance, NINDS does not have a targeted TBI set aside, right? We don't set aside money just for TBI. We expect and hope that you guys will submit great grants, and if you guys submit so many great grants that our institute could be the TBI institute pretty soon, but that's unlikely to happen. As far as NIMH, so I'm not sure if Ferris, so I work with Ferris Tuma.
Susan's here.
Oh, Susan's here. So NIMH is obviously the lead for post-traumatic stress. They're the leaders, I think, in this, what we've heard about already today, in the innovative approach for our docs for understanding mental illnesses and including post-traumatic stress. And other institutes, though, that are involved in the RNA for the NRAP for psychiatric health, though, include NIDA, National Institute of Drug Abuse, and NIAAA. So they do participate, but they're smaller participants. So Colonel McGurk can probably tell you much more about NIMH's contribution to the psych health part of the NRAP. For TBI, I have, so if you go to the next slide, I actually have two of the same slides somehow, but I thought I made two slides. But so for TBI, one of the things that the NRAP drove was the development of FITBIR, which is, we talk about data sharing, and I hear data sharing all the time. FITBIR is actually an effort funded, the money came from the Department of Defense. NINDS helps, we support the infrastructure for it, and we provide personnel to run it. And what we do is the DoD and the NINDS, not NIH, I'll note, requires all clinical investigators to submit their data to FITBIR and share their data within a year post after they're finished. I think that the NRAP and what the Cohen Veterans group could do is helping with data sharing is accelerating data sharing, because we talk about accelerating the process, accelerating the learning, but for instance, I just funded a 7-year project, so that project is going to share its data in 2025. That's not really an acceleration, right? It's data sharing, but it's not acceleration. So FITBIR is, and FITBIR is starting to take shape, and there's lots of data in there. There's the PROTECT trial data set in there, which is a progesterone trial on severe TBI. It's available freely. You can get an account, and you can download it, and you can reanalyze it, and you can confirm your hypotheses with those data sets. But we have other wins. Let's see if the next slide is the same slide it is on here. Okay, so the next slide was supposed to be one of the biggest studies that NINDS invested in for TBI is called TRACK TBI, and many of you, all the TBI people here probably know about TRACK TBI, but it's really an observational study to get a heavily phenotyped group with imaging, outcome data, across severities from mild to severe TBI and follow them across for a year. So we get measurements acutely, we get them at 6 months, and we get them a year. What NRAP did, and part of that is the brilliance of Colonel Hack, is took NIH, like I said, is investigator initiative, so that came through an R01 mechanism. And we can't really direct where the next steps or where you can hook your wagon to track very well at NIH. But the DoD can. So the Department of Defense, under Colonel Hack's leadership, developed what Stu was talking about, which is the TBI endpoint development. So they take the data, they are closely associated with track TBI. They take the phenotypic data and they start to test hypotheses and develop which endpoints are most important, which are going to be the most promising ones to bring to the FDA. The DoD is also invested in extra money called Track TBI DoD. So we're working together on those to accelerate these processes. And that all that data is eventually going to be into FITBIR and shared in FITBIR. And it's the last thing that NRAP has brought in for data sharing is that Stu is, the other success story is the Sensei, which I think Stu's probably going to talk about. But Stu has pushed forward the VA to actually share data in FITBIR. So Sensei is the first study that the VA study that's going to be put into FITBIR. So this is a true success story for the NRAP is centered on FITBIR and data sharing. There's a long ways to go, and we can talk about some of the things we still need. And Colonel McGurk talked a little bit about it with particularly biosamples. But we made significant progress, and we're leveraging each other's strengths to help accelerate TBI research.
Thank you. And with that, why don't we go back to slide 74, which I believe is Stuart's slide. Yes, thank you. And you can pursue that and explain what Sensi is. Okay.
Sensi is the Chronic Effects of Neurotrauma Consortium, and it was a DoD VA a collaboration that started with people of like minds, Dr. Hack and myself, where we both had our ideas. I had an idea when I first came to the VA that TBI is not just one hit to the head. It is probably a accumulation of a lifetime of insults through maybe abusive family life, contact sports. If you go into the military, you have basic training, combatives, combat, and it's repetitive over time. And so we, VA had an RFA back in 2011 to start looking at chronic effects of neurotrauma. And then Dallas also had the idea over around the same time on the DoD side. And we were able, thanks to our leadership, and the NRAP, to put that together that formed the basis for the one of the two consortia that the NRAP called out for. So the Sensi was one. The consortium to alleviate PTSD, with Colonel McGurk can speak about, were actually called out in the NRAP. And so what this is a sort of like the Framingham type study for TBI. It's looking at multifactorial issues associated with TBI to develop diagnostics, to develop looking at determine whether repeated brain injuries can lead to neurodegeneration, and also the comorbidities that tend to be associated with TBI, such as not just cognition but mobility, sensory, which also includes pain, and endocrine issues, as well as psych health. It appears that TBI probably decreases resiliency and increases resistance to treatment and also the chronicity of it. And then the one part that we have not gotten to yet in the NRAP is actually development of treatments. But we're trying to understand what are the chronic effects of TBI. What's causing it? Is it inflammation? Is it other environmental exposures? Is it taking maybe opiates? Who knows? But we are working on those right now. Next slide, please. And so the Sensi has really grown over the last 4 years since 2012. and it includes these sites across the country. As they're both VA, academic, and also DoD. And I guess good. Let's stop. Okay, hold on. So not only these 10 studies are not only research studies, but there's actually one development study, which is developing the technologies for a phantom for diffusion tensor imaging. Because one of the aspects that we have, not only don't we have a biomarker for detecting brain injury, but we have no way of monitoring the atrophy that occurs after brain injury over time, especially to the white matter. So the development project that we've been funding through Sensi is a phantom to calibrate magnets, MRI machines, not only across different machines, but over time. So if you have a Framingham type study where you want to look at TBI as a chronic condition and what kind of atrophy takes place with TBI, you need to be able to calibrate that machine so if you can accurately detect that degeneration that may be taking place over time. And if you develop a treatment, you want to know whether you're slowing down that degeneration. Okay, next slide. And so when it comes to VA, looking at long-term vision is keeping the focus on that and preventing overcoming distractions that may come in the way that often occurs in DC. TBI also is establishing TBI, especially looking at TBI chronically, as a discipline within neuroscience and in medicine. And also help working together with private industries, such as on the Phantom, to increase investment in TBI, and also communicating that TBI not only disrupts cognition, but has all these multifactorial effects. As we mentioned before, classifying TBI and developing diagnostics and outcome measures. Next slide, please. Okay, let's go next slide. One recommendations. Since TBI is a chronic condition, you need to have patience. You can't find overnight therapies and treatments for something that is a chronic disease. And also, as mentioned before, and Dr. Marmer's talk and Dallas, that there's a whole host of issues that come into effect when you talk about TBI. Everything from genes to sex differences. Sharing of information, what Dr. Bellgowan was saying, and without, which is important to our veterans because they were in battle. The populations that they fought against are still around, and risking re-identification of these people not only may endanger their safety, but also their privacy. So sharing information without risking re-identification is very important. And developing new tools for studying TBI, which is great teamwork between DoD and NIH, and helping the cooperation better across all the sectors. Thank you, and stakeholders. Next slide.
Thanks.
Okay.
Great. Thank you very much. So it's clear that, again, that there's been a tremendous amount of interagency interaction and coordination, and that now in those first early years of the NRAP program, we've been able to establish a number of programs that are building the biomarkers, the fundamental foundational information that we're going to need to advance the field, especially in the diagnostic space, and certainly now even some very innovative therapeutic approaches. I'd like John Krystal now to tell us a little bit about where we need to go now in terms of our therapeutic targets. So, Dr. Krystal.
Thank you. Thank you, Magali. It's wonderful to be here and to be part of this discussion about the national effort to address the problems of the returning soldiers and warriors and veterans who have come back with PTSD. I'm John Krystal. I am part of this wonderful consortium called the VA National Center for Post-Traumatic Stress Disorder. Terry Keene leads another division on it devoted to behavioral science. I lead the part of the National Center mission devoted to neuroscience and medications. I think that there is a national challenge or crisis related to the development of medications for post-traumatic stress disorder. And I think that there are real opportunities at the moment to try to tackle this crisis by trying to engage interested parties in the VA, Department of Defense, pharmaceutical industries, and incredible new initiatives like Cohen's Veteran Biosciences to try to address this. Part of the issue is that our knowledge of the neurobiology of PTSD, I would say, is rather limited, but it's getting to the point where we're being able to identify circuits and molecular mechanisms that might be targeted. And it's interesting that across town today, VA is holding a meeting with several pharmaceutical companies to try to figure out how to build these collaborations. So as was alluded to in Dr. Marmer's presentation, we're in a real conundrum, really, in PTSD. In VA, we have over 600,000 veterans who need treatment for PTSD. And all we have to treat them with in terms of FDA-approved medications are two medications, Zoloft, sertraline, and Paxil, which are practically the same medication. And so these are the only medications that we've had now for over 15 years, approved by the FDA. And there are, I'm on slide 90, there has been relatively trivial investment in post-traumatic stress disorder by the pharmaceutical industry, certainly not proportionate to the nature of the problem or the scientific opportunities. In fact, there have also been very few tests of new medication strategies for PTSD funded by NIH, funded by the Department of Veterans Affairs, or funded by the Department of Defense. There is not a huge group of people writing the grants that we need to advance this field. And so part of the work that we have to do is to think about how we're going to develop that workforce, and I'll mention that again. The result, as alluded to by Colonel McGurk, is that the typical patient with post-traumatic stress disorder treated in the VA is receiving medications that we have no idea about the risks and benefits. Many patients, particularly with chronic PTSD, are treated with 3 and 4 medications, some of which, as Dr. Rothbaum, who's here today, has shown, impede fear extinction. So we need to really understand what the medications that are currently prescribed are doing, and we need to make sure that there are new medications being tested for PTSD. So on slide 91, let me just reiterate what we need. Definitive clinical trials for the most commonly prescribed medications to establish their risks and benefits. These could be supported by consortia of interested parties like Cohen's Veteran Biosciences, VA, DoD. We need early phase clinical trials of novel mechanisms that have been identified from both basic and translational neuroscience studies. And there's a real opportunity for partnership there. And I believe we need an investment in development of the people who are capable of doing this kind of research. I would say from a 10,000-foot view of the field of PTSD, that psychiatry as a whole has been relatively underrepresented, particularly in the neurobiology and pharmacotherapy areas. I'd like to move to slide 93, which is titled, Where Will We Get New Targets for PTSD Treatment? I think that we are learning a lot from basic animal neuroscience research, and we're going to have a lecture from Dr. Kandel that I think will help to make that case. But translational neuroscience, these are the studies that are conducted either on human tissues or using neuroimaging research, are, as have been alluded to, are helping to identify circuits and molecular mechanisms that could be targeted with behavioral therapies, biofeedback kinds of therapies using neuroimaging, brain-machine interfaces, neurostimulation treatments, as well as medications. And there are so many new areas and exciting areas where this kind of work is going on that I think there's tremendous potential. Drugs, as you know, work on molecular receptor targets in the brain. And so it's critical that we have a component of this research identifying or helping to guide the identification of those molecular targets. And in a slide that doesn't have a number on my page, it's called Example 1, targets from PET imaging studies. I just highlight that there are now a growing literature of studies of looking at various molecular targets and how they're altered in association with PTSD. Studies of the norepinephrine transporter, serotonin receptors, GABA receptors, cannabinoid receptors, that's where marijuana and its constituents bind in the brain, glutamate receptors, and molecular targets for inflammatory signaling in the brain. A growing set of these targets that I think could be used to either be biomarkers of treatment or help to themselves be targets for novel therapeutics. And I'm going to highlight one area that's of high mutual interest between the National Center for PTSD and Cohen Veteran Biosciences, and that's illustrated in slide 92, which is titled From Molecular Signature to Treatment Target, which is the first opportunity that we have now to actually analyze postmortem tissue from people who have had PTSD and to look at in that human postmortem tissue for the molecular and cellular signatures of PTSD that will give us a much deeper insight. The VA has established a national brain tissue repository, which is based at the Boston VA, and Cohen Veteran Biosciences is establishing a parallel synergistic initiative under the leadership of Dr. Carrie Ressler, who is here today in the audience. And I thought I would share the first result of that analysis of postmortem brain tissue led by Ron Duman in our division and at Yale University. Looking at a region of the brain implicated in mood disturbance, which is called the subgenual prefrontal cortex, in postmortem tissue of people with PTSD, the strongest molecular signal to arise, and one that has now been replicated in a larger database, is a reduction in the level of a transcription factor called SGK1. I'll leave off the details. It's probably not that relevant to many of the people here today. So a reduction in this transcription factor, SGK1, in the frontal cortex of people with PTSD. The big question you always have when you have these kind of molecular findings is, what does it mean? So the first question you might ask is, in the reduction is illustrated in the figure to the far left. The next figure to the right in that figure, I can't point to it because I can't even see the figure from where I'm sitting. But the next figure to the left, to the right of that far left figure, shows that if you stress animals and expose them to uncontrollable stress, that it reduces the expression level of this signaling mechanism, SGK1. Okay, so it's reduced in humans who've been exposed to traumatic stress. It's reduced in animals exposed to something like traumatic stress. What happens in an animal who is not stressed if you reduce the brain's ability to express this transcription factor? So a genetically engineered animal is created that doesn't express very much of this transcription factor, SGK1, and lo and behold, these animals look like they have PTSD, even though they've never been exposed to stress. They're hyper-reactive to stress. They have all kinds of other related anxiety, mood regulation alterations. That raises the next question. Okay, what if you could abnormally increase the level of this transcription factor called SGK1 by overexpressing it in the brain of animals? Lo and behold, on the figure to the far right side, you find that if you overexpress the level of SGK1, that animals now become resilient to the effects of this kind of animal model of inescapable stress. So here you go from a molecular signature in brain tissue to a potential intervention of raising SGK1 levels. And we're now actually testing in the animal models, got some preliminary clues about medications that can raise SGK1 levels in the brain. So you go from molecular signature to possible candidate pharmacotherapy for PTSD. And this is the kind of opportunity that we have as a field right now, which is to capture the incredible explosion in translation, basic and translational neuroscience, and bring it in the service of people who have PTSD. This is the first time that we have this opportunity, and it's one I'd really think that we can all partner on in capturing.
So just very quickly, what I mentioned in the pathophysiology of PTSD meeting in June, I'd like to have it now, because the juice is flowing a little with what Dr. Krystal just described. If it's got you wanting to talk more about that, please be on the lookout for that meeting, because that's exactly what we'll be talking about. So, and it leads to what the other thing coming out of the White House is the President's Precision Medicine Initiative. This is where we need to go. What works for who and why, so we can target it to the right person at the right time, the right treatment.
Yeah.
So I thought I was going to have a challenging time getting everybody to talk about gaps and opportunities and so on, but I think it's just been a fantastic, honest assessment of where we are now, where we need to go, and I'd like to open it up now to our audience to ask any questions that you'd like to ask of our panelists. I think we have about 5 minutes for questions. Is there any out there? No one has the question? Yes, we do. Okay. Dr. Kelly.
Hi, everybody. Jim Kelly. Is it on? Okay, thanks. What role, if any, might there be for an advanced neuroimaging such as PET MR? There aren't very many so far. PET CT has been kind of in the, in the game for a while now. But if you look at the resolution of at least a 3T MR, does PET, especially if we have, let's say, a tau-specific ligand, and I haven't heard discussion about that, but you folks know about that, is there something meaningful in advanced neuroimaging technology that's right around the corner, or something we should be pushing for?
So, I can answer part of that. Yes. So that's what so most of you probably know NIH has the BRAIN Initiative, which is about developing tools to go from look at systems from molecular and redefining cells all the way to ways to discover how circuits work. And one of the things that we don't know, one of the big challenges, as you're mentioning, in TBI, particularly the chronic effects of TBI, is being able to follow people. We try to match MR scans with PET scans, and simultaneous imaging may be a big help. So we're, as you know, NINDS is heavily invested in CTE, and we're trying to work backwards from that. So we developed first a brain bank, and we're funding the BU group to start to understand the pathophysiology in tissue, and then we funded another group for probable and possible, and has heavily based on PET imaging. But one of the things I think the Cohen Veterans Biosciences could really contribute is that we can't. So I have the great pleasure of also being the director for NINDS's brain bank. So we can't form registries. So they could form a registry which could help. So NINDS has a series of 6 brain banks where we have tissue available for anyone who wants to work on that tissue. Mm-hmm. One of the challenges for TBI, though, is if we want to follow people chronically to get that tissue after we've imaged them, we can't form that registry. But
maybe I could just comment on it also. So we do PET and we do MR. And that means that we get the PET on one day and we get the MR on another day. But if you have the main advantage from my perspective of the PET MR is if you're asking a question that where you would want to know what is the brain doing at the same moment or in the same time that you're doing that you get from the MR, what is it doing at the same time you get your PET measure? And that question is best answered if you can get those two kinds of measures
simultaneous acquisition. So, and that's very important, I agree. The other piece of this is, many of you know about Dan Pearl's work and the gliosis of blast injury, and even though it was just a series and not, the kind of hard science that so many of us would really push for. Those were all invisible on every kind of neuroimaging that was possible. And so I think we need to understand better why something's visible in blast-injured brains on pathology, much like we see in CTE and so forth, even though it wasn't really CTE. It was a different kind of gliatic scarring. We need imaging to actually see that in vivo. And I think that that's one of the big things that hopefully all of you that are working on this can push for as well. Colonel, in particular, thanks.
Thank you. I think Dr. Yes, Hoffman Officius has a question, and I just want to thank you for the comment about the brain bank. Tomorrow, Dr. Sabina Sabaretta will be presenting on the new Cohen brain collection, and I think this is something that we're looking at, so it's a great recommendation, and we'll certainly take that forward.
Yeah, thanks.
So I just want to point at two critical aspects of what we have heard about. One is that the experience from the neurodegenerative disease field tells us that treatments in the mouse do not easily translate into treatments, successful treatments in humans.
Right.
Effectively, none of the Hugo Gertz is listing more than 200 different treatments that did not work out. So just, with all the excitement about something that works in mice, be careful. It may not really, end up with the service people. So that's one of the things. And the other thing is more, operational. I was involved in discussions on the Global Action Plan against dementia and so on, and there was all this talk about sharing data. And that is an important aspect, but at least as important as sharing data is having shared semantics describing the data. And I haven't heard really about that so far. And I was wondering what your take on shared semantics is.
Actually, yeah, go ahead.
So for FITBIR we use, so NINDS has, and NIH in general, NIMH has a heavy database of common data elements also. So for the DoD, for Sensei, and for the NINDS studies, they are required to use common data elements, which is this common language, and metadata on top of that. So you can describe analysis and imaging with metadata also. I mean, that's one approach. There's multiple approaches to this. Spinal cord injury, for example, and ours is taking a very different approach, which is more the, create metafields that do it.
But the imaging is a good example of actually a lack of structured terminologies describing features that you would compute from images.
But yeah, so we've tested, part of what Colonel Hack put together in the TED initiative is, so TRAC has scanned 1,600 TBI patients so far, right? Well, not really that many, 600 because they're not all scanned. But then they brought through and they took our common data elements, went through double-blinded studies with radiologists to have them validate the CDEs, which needed tweaking. We tweaked them, and now they're coming back out, and they should be published. I think they already submitted the manuscript.
I just want to, you're talking about the mouse situation. With Sensei, there's only one study that is using either postmortem or preclinical studies with animals. And that, they're taking a unique approach, not looking so much at these transgenic models, but also looking at the role of, like, let's say, inflammation has on generating the chronic neuroinflammatory generating the chronic degeneration that's taking place in these animals. And at the VA, the animal studies that come through my panels are all chronic studies. So they're not doing anything to the animals after injury. They're waiting about 6 months or so at least, and then looking at the brains for signs of pathologies that are similar to what you see in humans.
I wanted to comment on the translation issue from animals to humans as well. I think to some extent we haven't done that in a very scientific way. We did, because I was responsible for the TBI part, we did an analysis of the 35, whatever, failed trials in TBI that didn't translate. And there were just fundamental flaws in the translation that were really the biggest cause of that. When you do your 200 animal studies, all involving a visual memory test, and then you do your human trials based upon a consciousness scale. I mean, and you do your outcome measures that are based upon a disability score, it's a completely different domain than you did all your animal trials. We've just done a bad job in the translation. If you're going to do a visual memory test in an animal, why don't you do a visual memory test in a human if you're trying to do a translation between the two? It's just some real fundamental flaws, and that's part of what the TED is trying to get at, is how to actually do that translation.
Yeah.
It's just, just you look, you do the rest, we've interviewed the PIs, they all realized that after the trials failed, that they didn't do the translation properly.
But the exciting thing that Dr. Krystal presented was its reverse translation, right? So you
Yeah.
found it in human, went back and recapitulated it in
That's
mouse.
the stuff that needs to
And that's like to me, the translational component.
But we have been there in
Dr. Kent.
neurodegeneration as
And
well,
Alzheimer's,
with the reverse engineering. So that is not the ultimate.
No.
Anyway.
Yeah, so I want to pick up on that theme of construct validity that we need to preemptively address across all of these animal modeling programs. Tomorrow we will hear about AMP It Up, which is being co-led with Dr. Carrie Ressler and Dr. Israel Liberson, who are here with us. And AMP It Up is focusing on PTSD translational models and developing construct validity so that we can address this head on. And I also want to mention that picking up on the theme of common data elements, we just launched last week with the FDA and the NIH the establishment of CDISC standards, which are coding standards for all of these tools, so that when they're entered and logged into these computer systems and these different databases, we're all coding the data in the same way.
Yeah.
That's what's going to facilitate our ability to integrate the data at scale. And so we're working very closely with our colleagues to convert all of these to a common standard as well. Another number of questions. We'll start with Dr. Evans.
Hi, I'm Alan Evans, McGill. A comment quickly to respond to the earlier question about what's on the horizon. There are new technologies that have been around for a few years now that allow you to do PET and MRI simultaneously. So you can deal with that issue of disjoint PET and MRI. The second point I'd like to mention is there's a lot of very elegant work around these days that examines network organization in the brain, structural and functional connectivity. Mm-hmm. And those kinds of metrics are likely to be closer to the behavioral deficits that you see in PTSD and TBI than simple focal measurements of did this piece of the brain get bigger or smaller, did it have a slightly higher or lower functional value. The network organization, I think, is going to teach us a lot over the next few years. It's far from a closed and understood field, but it's rapidly developing in many different areas.
Thank you. I think Mary here.
I'm going to bring it up to a layman's level because I'm nowhere near at the level you all. When you're going down this path, if you could please consider also secondary PTSD. I deal with it on the end of a caregiver, and that can range from the person who's at home to our men and women in the hospitals. And I've seen devastating effects to it ending up in suicide. And if we don't incorporate that language now, trying to incorporate it as an afterthought later, it doesn't have the same.
Yeah, I think that's why I mentioned that we fund research in family health and well-being, because it wasn't specifically called out in the NRAP, but we know from lots of data and just talking to service members, what's going on at home affects them, what's going on in the work affects home. So we're not neglecting it.
Exactly. And if you don't have a healthy home life, you're not going to have a healthy service member. And we can push that out further beyond the military. I mean, I think what you all are doing it's going to have much greater effects.
There are also, you might be interested to know, there are some medications that are being tested for PTSD that aren't addressing the anxiety or the flashbacks, but rather social cognition, social function, and even testing these medications as adjuncts to couples therapy in PTSD. So we're used to thinking about a drug as something that reduces a symptom, and we're now taking a much broader view of that, which could be very important.
Which is great. And I would also say not just have couples therapy, it's the individual. This person doesn't have to be being seen with the person who's had the brain injury or the injury or whatever.
Sure. Yeah, the VA has an active research program in that area where they fund research on families. They found some very interesting results from caregivers who are taking care of veterans of brain injuries and the effects that has on their health, not just mental health, but also their physical health.
Yeah, and thank you for that. I'm working with it on a daily basis, so I see it up front. And
Thank you, Mary. I think we have time for one last question before lunch.
Thank you all for being here. You kind of answered my question, so I'll just make a comment. I'm a 20-year Army reservist. Recently came out of a tour at Walter Reed, providing service to warriors in transition. There was a bullet that came up earlier that I note down, and it says, providing interventions to high-risk groups at high-risk times. And I just want to share with you out of my experience, please let your research be guided by what's most important to the soldiers, which is not necessarily finding a cure to an unavoidable physical response, but more so managing the experience of PTSD. And thank you for that, and thank you all for being here today.
Thank you all. I applaud that sentiment. So thank you for the panelists.